Hi guys. This is the video that I promised you guys after Walter Peters last week, where essentially I was going to take you guys through the complicated process known as cytokine storm. Rather than kind of messily creating this as I wrote, I decided to keep the image from your faculty guide and kind of talk you through it, also pointing out the important parts and the not so important parts. So remember, there's a couple things that we can kind of keep in mind when we think about cytokine storm. First off, the immune response is not always the good guy, okay? Sometimes immune responses need to be ended. And we talked about this a lot in your case yesterday, Camera Thompson Part 1. And in that case, really, we're beginning to see how to end the immune response. And part of the reason for that is all of what's happening here, okay? So we need to keep immune responses carefully controlled. Two, as we saw in Katie Diaz with EBV and CMV, many microbes are very skilled at immune evasion, okay? Now, I wouldn't necessarily say that what happens in a cytokine storm is a form of immune evasion, but it's certainly a form of immune manipulation, okay? So I want you to think about how this could potentially work. Much of what we're talking about in the context of cytokine storm that we discussed it are hemorrhagic fevers, okay? And kind of the crux of the conversation was, how do we go from a viral infection that infects a myeloid cell to a patient hemorrhaging out or bleeding out, okay? And how is this helpful to the virus? How does this lead to immune manipulation? Well, actually, the answer is very clear. It leads to new targets, okay? If the virus is in the blood, if there's high viremia, then anytime the blood leaks out of an orifice, be it, your eyes or your nose or through vomit or in the feces, you're creating a opportunity to encounter a new healthy host. So the advantage to the virus is very clear. So let's talk about the role our own immune system plays in these viral infections and why this leads to the phenomenon known as cytokine storm. So really, when I use the term cytokine storm, it's a really great example of what I think most of immunology is built on, and that's the concept of a positive forward feeding feedback loop, okay? So a great example of this that we'll actually use here is that Th1 cells produce interferon gamma and IL-12, which in turn makes more Th1 cells, which produce more interferon gamma and IL-12, which in turn makes more Th1 cells. So the immune system is designed to keep itself going until such a time that something else, something new, like a T regulatory cell, comes in to stop it, okay? And in the case of hemorrhagic fevers, that control doesn't happen. So we just keep having this forward feedback loop of cytokine storm, okay? So let's take it all the way from the beginning. Remember, one of the goals of this case was to enable you to realize how much you really have learned already about the immune response. So in this case, I was using a patient with dengue fever. And I think this could actually pertain to almost any of the hemorrhagic fevers. So if we just kind of ignore the dengue fever or dengue hemorrhagic fever component for the moment, let's call this instead, I don't know, Ebola. All right. That's always a fun one. All right. So Ebola is going to come in and infect these myeloid cell targets. So that could be anything. It could be monocytes, it could be macrophages, it could be dendritic cells. It's any of these myeloid cell types. What we know is that it's not epithelial cells, okay? So this little red guy is my virion. And it gets picked up by its receptor, and it gets into my monocyte population. Inside the monocyte population, it begins replicating itself, creating more virions. That's what viruses do. They replicate themselves. Now, there are going to be a couple things that are going to happen internally here. Let's make note of that, okay? As the virus is attempting to replicate itself, we are going to get type 1 interferons turned on, okay? These type 1 interferons are interferon alpha and interferon beta. Type 1 interferons are really great at actually controlling nearby cellular responses. So they basically tell nearby cells to increase expression of MHC class 1, decrease protein production, all other proteins, and decrease endocytosis. So it's basically a batten down the hatches and show us what you got pathway, okay? So that's kind of our first step. But it's not enough to completely stop the immune response, all right? The immature dendritic cell is going to pick up the virus and take it down to the lymph node. And as it goes, it's going to mature. When it gets there, it's going to present this viral antigen to CD8 and CD4 positive T cells. The CD8 positive T cells will basically be benefited by the presence of the CD4 IL T cells because the CD4 T cells will produce the IL-2 that the CD8s need to proliferate and grow. These CD8s will go out into the world and they will kill virally infected cells. As this cell dies, nearby monocytes and macrophages will not only pick up dead cells and become activated themselves, producing cytokines, they will also pick up virions and potentially become infected. So we're promoting the immune response in two ways, through infection by these cells picking up virions, but also through activation as these macrophages that love to eat dead and dying cells start producing all of these cytokines. So that's one arm of it. Basically, the innate cells, your macrophages and monocytes, are producing cytokines. They're going to produce things like IL-12 and TNF and IL-6 and IL-1 beta. And hopefully at this point in your immune knowledge, you know that these cytokines are super potent and they do things like create a leaky endothelium. So this has now been perpetuated because we had the first time the cell was infected that these cytokines got produced. Then we get this cell dying or lysing or virions coming out of that cell, and we get more of these cytokines being loosed. We're perpetuating the problem. We added in CD8s. We also added in Th1 cells. These Th1 cells are going to produce their own interferon gamma and IL-12. So now we're increasing the response even more. We have even more interferon and even more IL-12. This interferon gamma, do you remember what it used to be called? Macrophage activating factor. So that's even further activating the macrophages. So the net result is we're now activating almost all the cells in the immune response, and we're making this kind of cytokine soup, okay? So we're also getting activation of NK cells, which are great for killing infected macrophages. But remember, every time we kill an infected macrophage, we could potentially be producing virions, and also we're activating more macrophages because macrophages love to eat macrophages. So basically, we're just creating this soup of cytokines that is exacerbating and amplifying the immune response over and over. So each time it goes around, the magnitude of the immune response gets bigger and bigger. Okay, so I'm making my arrows bigger and bigger. Okay, and that's basically what leads eventually to this damage. All right, so that was if it was Ebola. Now, what if I get rid of all of this? All right, and we talk about what happens with dengue fever. So the first time around you get dengue fever, you're going to feel pretty bad, okay? I mean, that's no walk in the park. There's a lot of pain, there's rash, conjunctivitis, all sorts of ugly things. And yeah, you're going to have some weakening of your endothelium, which is going to lead to some mild edema and inflammation that we expect with fever, okay? But here's something we didn't talk about with Ebola that still probably would happen. In fact, it does happen. It's how we get convalescent serum. These Th1 cytokines, IL-12, interferon gamma, they basically promote B cells to become plasma cells. These plasma cells secrete IgG, okay? Now, this IgG is a good thing. If it's Den1, let's say, and we encounter DEN1 again, this IgG is a great neutralizing antibody. So it's basically going to stop the virion from being able to bind to its target receptor, which is DC sign, and actually be able to get into the monocyte macrophage. Here's the bad thing. It's not a neutralizing antibody for any other serotype. So if it's DEN2 through 4, we're no longer getting that neutralization, okay? So now it's doing something very favorable for the virus. It's still going to bind the virus, but not in such a way that it neutralizes the virus. Instead, what it does is it basically becomes a virus helpful opsonin. So remember, anytime you have an antibody, you have this Fc portion. So for an Ig molecule, this is Fc gamma. And on macrophages and monocytes, we have Fc gamma receptors, okay? So basically receptors that recognize this portion. So basically, then the virus hitches a ride on the antibody into the cell it wants to get into in the first place, and that means that many macrophages get infected that much faster. So we're still going to go through this whole process, but it's going to be enhanced and amplified, and it's going to happen faster. In this case, our memory response is actually promoting the faster accumulation of cytokines. And that's what actually leads to hemorrhage, because we actually build up this cytokine storm, this huge forward feeding loop, so much faster than our body can actually handle it. And that's what leads to hemorrhage. So that's kind of the whole story of how we get from one side of this, where we're just infecting cells, to the other. So let's just go over kind of a couple of rules of thumb that I think would be helpful for you to keep in mind as you're preparing for your exams. So for right now, there are 3 cell types that I kind of expect you to be able to make. These 2 for the formative, and by the time you get to the summative, probably Tregs as well. So for Th1, your main cytokines are IL-12 and interferon gamma. And they're produced by Th1 cells, and they also make Th1 cells. For Th2, it's IL-4 and IL-10, and they also make Th2s. For Tregs, IL-10 and TGF-beta, and they also make Tregs. This leads to largely production of IgG. This is largely going to lead to production of IgE. Here, we're not producing antibodies. We're not trying to start an antibody response, because antibodies actually promote inflammation. Instead, what we're going to do here is that if we induce a Treg, we're actually going to stop the responses that are made by Th1 and Th2. Conversely, if a cell has decided to become a Th1 cell, it won't become a Th2 cell, and vice versa. Now, there is some confusion on this in the literature, that cells are a little bit more plastic. But for our purposes, what we're trying to understand is we're trying to understand the pro-inflammatory environment, or you may have also heard me call it the cytokine milieu. We're really just trying to understand the environment in which these T cells were made, okay? Now, I've heard some students mention their confusion about IL-2. IL-2 is important for all of them. It's especially important for making Tregs, but remember, all lymphocytes need IL-2 to proliferate and grow. So if you can understand this, and then when you look at this picture up here, you can have the general understanding that we're going from the innate immune response, which begins with things like IL-1 beta, IL-6, TNF alpha, and that they have their own effects, be it fever, vasodilation, and vascular permeability, which leads to edema, into the adaptive immune response, which in the case of viruses is largely promoted by interferon gamma and IL-12, and that this actually feeds back to this, just as this feeds into this. You can see that we begin with the innate, we recruit the adaptive, and then both the innate and the adaptive continue until such a time that the pathogen burden has fallen down and the immune response can be safely ended. That's kind of where you should be at this point, okay? Don't worry if you can't explain it in quite as much detail, but these are the major themes I want you to be able to take away from this. Good luck, and let me know if there's anything else that you need.